that is just as true for bacteria as it is for people, as it is for plants, as it is for fungi. Okay, so this metabolism chapter is very similar to material that you would learn in general biology or anatomy and physiology. We're back to that for a chapter material that you may have seen before. But the bottom part of this slide says why we're doing it. There are some differences between bacterial metabolism and human metabolism. And just like in the cell chapter, we're always looking for differences between us and them, right? So we can have antibiotics target bacterial ribosomes, but not human ones, right? Ones that destroy 7DS, but not 80S. Antibiotics that affect peptidoglycan because we don't have any, right? So we want to know the differences between their metabolism and ours to give us possible drug targets for antibiotics. So, going back to the top here, metabolism is all of the chemical reactions that happen in a cell. As humans, we are super focused on just the part of metabolism that burns fat, right? That's all we care about, all those commercials about boosting your metabolism and trying to lose weight. We're focused really on the anabolic reactions, right? Doing work, right? Using energy from the food that we have or the food that we've stored to do something. Move the cell, make molecules, active transport. We got the other half of metabolism, which catabolic reactions. We're going to take energy that we've taken in and convert it into a usable form. So we're going to take the energy from food, turn it into ATP, then we're going to use that ATP to do work. So that's sort of two halves of the equation. Good with those basic definitions? All right. So, energy, the ability to do work, definitions that you would get from a physicist or a chemist, right? Energy is the ability to do work. Two kinds of energy, potential and kinetic. Potential energy is stored energy. Kinetic energy is the energy of motion. So, if I took this ink pen and I hold it above the table, that's potential energy, right? It's got energy stored in it because if I drop it, it's going to fall, right? And that's kinetic energy, energy of motion. Potential stored, kinetic while it's moving. Bless you. Applying that to living things, all living things, potential energy is stored in bonds. Whenever we drew a line between two atoms, that bond has energy stored in it. That's why we in bacteria eat carbohydrates and proteins and fats as food, because there are lots of bonds in those molecules, lots of energy stored in them. So when we want to get some energy out of it, we have to break that bond, separate those two carbons from each other, and that will release energy, which we can use to make ATP or do some sort of work like contract muscle, do active transport, right, whatever we need to do. So all of our potential energy as living things are stored in bonds. So that brings us to the first law of thermodynamics, which I'm sure you've learned in one form or another since probably somewhere around 3rd grade. Right? Energy is neither created nor destroyed, only transformed. At least that last part of it should sound familiar, right? The whole created nor destroyed, only transformed. What that means for us is that energy is constantly going back and forth between kinetic, potential. So right now, energy is traveling through space from the sun, right? Light and heat from the sun are crossing the universe to get to us, right? So that's kinetic energy right now. That energy is moving through, it moves in a wave-like pattern, right? Light travels as a wave, hence the wave motion I was making there. So that energy is waving across space. Once it hits the plants outside absorb that energy and use it to make sugar, right? So it's stored in chemical bonds. So then it's potential energy. It's moving across space as kinetic. The plants use it to make sugar. Now it's potential. We eat the plants, break the bonds. Now it's kinetic energy again. We might store it as glycogen or as fat. Now it's potential energy again. So it's constantly going back and forth. Kinetic, potential, kinetic, potential. Does that make sense? All right. Chemical reactions. Since we're using chemical bonds as our source of energy, we're going to be doing lots of chemical reactions in metabolism. Got the basic one up there. A, B, C, and D represent chemicals, right, depending on which specific reaction we're talking about. A and B are the reactants. They're the chemicals present before the reaction occurs. And of course, it doesn't just have to be 2. Sometimes there's 1, sometimes there's 5. Okay, but they're always to the left of the arrow. The reactants are to the left of the arrow, present before the reaction occurs. The arrow represents the reaction happening. And C and D are the products, chemicals present after the reaction occurs. They're always to the right of the arrow. And again, there's not always two of them. Sometimes there's one, there could be three, there could be seven, depending on the reaction.
Arrow?
The arrow is the reaction happening. So you'd read it A plus B yields C plus D. The arrow represents the reaction. All right, so you apply energy to that reaction, A plus B yields C plus D. All chemical reactions really fall into one of these two categories depending on whether energy is being stored or released. Okay, so in an exergonic reaction, energy is released, energy is exiting, right? Exergonic, energy is released. The way your book says that is that there's more stored energy in the reactants. These molecules have more energy in them, and therefore we're going to release energy by breaking them down. So in that kind of reaction, this might be a basic one. This is glucose, the sugar made by most plants during photosynthesis. When we eat the plant, we're going to digest and break down that sugar and turn it into carbon dioxide, which we exhale, and water. And the way I know that this is an exergonic reaction is because of the number of bonds. We've got 6 carbons, 12 hydrogens, 6 oxygens, that's 24 bonds, right, that we have in this molecule. That's a lot of stored energy, right? Over here, carbon dioxide has essentially 2 bonds. If you've had chemistry, you know they're double bonds in carbon dioxide, but essentially 2. Water has 2 bonds. Right, we have the oxygen in the middle and the hydrogens hanging out like that. So we're starting with something that has 24 places to store energy, and at the end of the reaction, we only have 4 places to store energy. That means that energy is the silent product. Energy has been released in this reaction, because we've got more bonds on the left than we do on the right. Now also, if you've had chemistry, I know my equation isn't balanced, right? If there are 6 carbons here, there should be 6 carbons there. But this is biology, and we don't always worry about that kind of thing.
Question, do we need to know that?
No. I'm never
Okay,
gonna
you're just
ask
showing
you.
an example.
Right,
Okay.
I'm just giving you an
Just
example.
ask.
But does it make sense? Wherever there is the most bonds, that's where the energy is stored. We got less bonds over here, so energy was released, exergonic. Endergonic reactions are the opposite. This time we're going to store energy in the reaction. So when the plant made that glucose, they took carbon dioxide from the atmosphere, water they got from their roots, energy from the sun, and made glucose out of it. So now we're starting with things that have a small number of bonds and making something that has a lot of bonds in it. We've stored energy by creating a large molecule. Okay, so endergonic, energy is stored in the reaction.
And exergonic is releasing it.
It's released, yeah. Okay, so we're good with those? All right, even though we like to write these big equations, call this one photosynthesis, call that one respiration, chemistry doesn't happen in one giant step. It's not like plants are going to make all 24 of these bonds at once. And when we digest the sugar, we're not breaking all 24 of these bonds at one time to create water and carbon dioxide. It happens in a pathway. There's a series of small steps, little reactions that add up to this big equation we like to write. Okay? So metabolism doesn't happen in one giant step the way we write it. It's a bunch of little teeny reactions that happen one after another after another. And when you add them all up, it equals the big reaction we like to write. The reason it happens in a pathway is because it's more efficient that way. Right? If we took all 24 of these bonds in the sugar that we eat and break them all at once, so much energy would be released that a lot of it would be lost as heat, and the water inside of our cells would boil. We don't want this to happen, right? So we don't break all 24, neither do bacteria. They don't break all 24 bonds at once. They break 2 or 3 bonds. Release a little bit of energy, absorb that energy, make ATP out of it, then go back, break 2 or 3 more, release a little bit of energy, absorb it, use it to make ATP. We're able to absorb more of it, make more ATP out of it, hence be more efficient if we break a few at a time. That make sense? Willetini reaction. There are 3 types of pathways, Willetini reactions that can be used. The definitions are all here, and I'll come back to this if you want to write them down, but I think it's easier to look at the picture explaining. Alright, so the first one, linear reaction. The one starting compound, you always get the same end product, right? So when we digest sugar, we always make carbon dioxide and water out of it. Those are the same end products every single time. With a branched pathway, the one starting compound could be used to make 2 different end products, depending on what the cell needs, right? So if the cell has plenty of end product 1, then when we use that starting compound, we'll use it to make end product 2. And if they have enough end product 2 and they need more end product 1, they'll switch. So in that kind of pathway, there are 2 possible end products from the same starting compound, and the cell decides which one to make based on which one they're running out of at the moment. And in the cyclic pathway, the one starting compound is always recreated. It makes a cycle or a circle, and you always recreate the starting compound. And we're going to see examples of all of those in this chapter. Okay, so if you were copying definitions, there they are. Linear, the same starting molecule always makes the exact same end product. Branch, the same starting molecule could make more than one end product depending on what the cell needs. In cyclical, the starting molecule is always regenerated. You can see a couple of your copying, so I'm sure you have time to get that down if you want.
So branch doesn't just have to be 2 though?
No, it could be 3 or 4 or 5. It just depends on the pathway. You've got multiple end products. Anybody need more time? All right. Now what we're going to do is go through the ingredient list, right? If we're going to do metabolism, it's like if you're going to bake a cake, there's that list of ingredients you need to gather before you can make the cake. We're going to go over the list of ingredients, the components you need to make metabolism happen now, before we can actually talk about how to make the cake, how to do metabolism. So the first ingredient, bless you, first component is ATP. This one we've already talked about, so this should be reviewed. Adenosine triphosphate, the usable energy molecule of cells. Us and bacteria, we eat fat, we eat protein, we eat carbohydrate, but we've got to convert it all into ATP before we can use it. So, same picture that you saw in that last chapter with ATP when we're talking about active transport. You need some energy to do work, you break a bond attaching one of those phosphates, you break the bond, energy gets released, you can use it to do active transport or whatever work you need to do. And in this chapter, we're going to focus on the other half of this picture. You have adenosine diphosphate left after you do that, right? 2 phosphate groups attached. So now we need to get some energy from somewhere and reattach the phosphate to convert it back into ATP. Okay? So that's called phosphorylation, attaching the phosphate group onto any molecule. In this case, we're attaching it onto ADP. And there are a couple of ways to do it. There's your phosphorylation there at the bottom. There's a couple of ways to get the energy to do this, to put the phosphate group back on. So here's where the new information is we didn't talk about with active transport, where that energy comes from. So, 3 ways to phosphorylate to get the energy to put that phosphate group back on. The first is called substrate level phosphorylation. For this, the energy comes from an exergonic reaction, from breaking a bond. Okay, so we're going to eat the sugar and digest it, break some of those bonds in the sugar, release that energy, use it to make ATP. That's substrate level phosphorylation. I'm sorry?
I can't remember, but it's an easy way to remember that. I gotta find my notes on intro to bio.
Yeah. Okay. Like a mnemonic device or something that you had?
Yeah, I remember because I had you for like lecturing the other lady for lab, but she told me go ahead.
Well, if you find it, let me know.
I'm gonna go get it out my box.
Look for it.
Right.
Okay, so substrate level, using the energy from breaking a bond. Oxidative phosphorylation is going to use proton motive force. This goes back to active transport as well. Remember, bacteria are the only organisms that make proton motive force on their cell membrane. And proton motive force was a separation of charges across the membrane, where the outside was positive